Hi, everybody. Welcome to today's Hot Topic program, Recent Fossil Discoveries at Drimolen Cave, South Africa, with Dr. David Strait. My name is Briana Pobiner, and I'm a paleoanthropologist and educator at the Smithsonian's National Museum of Natural History. I'm a brown and gray-haired woman in a red sweater with a colorful necklace, wearing headphones in front of a Zoom screen with an African savanna photo with grass and an acacia tree behind me. Whether this is your first time joining us for a hot topic or you've attended before, we're very glad to have you here. Before we get started, I have a few housekeeping notes. This discussion offers closed captioning. You can turn the captions on or off via the CC button, which should be located at the bottom of the Zoom interface. We're in a webinar format, so we can't see or hear you. As you have questions, please go ahead and submit your questions to the Q&A box, which is at the top or bottom of your screen. It looks like two speech bubbles, so that we can sort through as many as possible. The Q&A time tends to really fly by. The Q&A box is also where we'll share any relevant links during the program, so keep an eye out there. We'll start with a presentation by our speaker, David Strait, and then I'll join him here to take your questions. During the presentation, I will also write answers to some of your questions, at least any that I can answer, as will another member of our behind the scenes team, Dr. Ryan McRae. So now I'd like to go ahead and introduce our speaker. Dr. David Strait is a professor of biological anthropology at Washington University in St. Louis, and he's a paleoanthropologist who studies several key aspects of the fossil record of human evolution. First, he's interested in understanding how and why the various species of early humans or hominins diversified. And a large part of this entails understanding the pattern by which these species are related to each other in an evolutionary sense, which is known as reconstructing phylogeny. Second, he's interested in the evolution of diet and feeding in fossil hominins, and he works with a large team of collaborators from many universities that use engineering, experimental, comparative, and ecological methods to test hypotheses about feeding biomechanics in fossil hominins and non-human primates. And finally, to discover new fossils to better understand human evolution, David has led and or participated in excavations in Africa, the Near East, and Europe. And today we'll hear about one excavation he's involved with, an early Pleistocene cave in South Africa called Drimolen that preserves evidence of robust australopiths and early members of the genus Homo. So now I welcome David to share his presentation.
Thanks very much, Dr. Pobiner. It's a pleasure to be here with all of you, and I'd like to thank Dr. Pobiner for the wonderful introduction and also the invitation to speak. Let's see if I can do this, share my screen, and hopefully that's showing up, and I am going to start. There we go.
All good.
Thanks very much, folks. I'm going to talk to you today about some really interesting and fun discoveries that we've made recently at a site in South Africa called Drimolen Cave, where I've been working for the past several years with a very large international team. But before I go into those details, I want to spend just a couple moments doing a brief recap of human evolution in a very broad sense, so that we're all sort of on the same page. So what we're looking at here is on the bottom, there's a time scale from the present day, 0, all the way on the right to about 8 million years ago on the left. And I'm going to then show sort of major events that happened in human evolution at various points during this time. So about 7 million years ago, we get the earliest evidence of fossil hominins or extinct humans who are more closely related to us than they are to chimpanzees. And these, there are a few species of them, but you might call them, broadly speaking, the pre-australopiths. And they existed between about 7 and somewhat less than 4 million years ago. And for the most part, these things are apes that walked on 2 legs. They were bipedal. That means they habitually walk around and stand on 2 legs. But they lack many of the very distinctive human-like features that we have, like a large brain, complex speech, advanced abilities to manipulate objects with your hands, and sophisticated tool use. About a little bit before 4 million years ago, we then find a second group of early hominins that appeared. These are known as the australopiths, and they are, in a broad sense, descended from at least one of the pre-australopiths. And these species still are largely ape-like, but they walk on 2 legs. But now they have added amazing adaptations in their jaws and teeth, and in their chewing muscles, that were probably adaptations for feeding on foods that were mechanically challenging to eat. From one of these australopiths, what is going on here? Why is my screen not advancing? There we go. Uh, uh, from these australopiths, a new group of hominins appear sometime between 3 and 2 million years ago, and we would recognize these as the early members of the genus Homo, that is our own genus. These species then begin to have many more of the traits that we consider very human-like. They have very human-like body proportions, the brains get bigger. They begin to make and use increasingly sophisticated stone tools over time. And then about 300,000 years ago, from some type of archaic Homo ancestor, we get the appearance of our own species, Homo sapiens. They appear in Africa about 300,000 years ago. These folks look just like us, very, very large brains, flat faces. And over time, they develop amazingly sophisticated tool technology, as well as an amazing ability to produce art, and therefore reveals advanced symbolic thinking. Okay, so that is just a broad overview of what has happened over the entire scope of human evolution. I'm going to focus in on this period here. Hopefully you can see my cursor. At about 2 million years ago, when we have both some australopiths alive in the human fossil record, but also early members of the genus Homo. And I'm going to tell you about our research at Drimolen Cave. Drimolen Cave is a site in South Africa. We run the site as a field school, so students can come and participate in excavations, learn excavation techniques. They learn about paleoanthropology, about geology and archaeology. Many of the students who come are college students, but we've had a high school student come. We have adult students come who are in the middle of their careers. We've had some retirees come. So you are all welcome to come and participate in the field school. We won't be running it in 2026. We're taking a pause, but then we'll be running it again in 2027. And I believe the link, if you want to learn more about the field school, has been made available to you. Okay, so where is this site, Drimolen Cave? Drimolen Cave is found in South Africa. Here's a map of Africa. There's the country of South Africa down at the bottom. And then within South Africa, just a little west and a little bit north of Johannesburg, which is sort of the major city in South Africa, there is an area where the geological conditions are excellent for both the preservation and also the discovery of fossils. In South Africa, this is known as the cradle of humankind. And each of these little dots on the screen here represent sites preserving evidence of human evolution. Why are all these sites here? It's because there is an outcrop of a particular type of rock. It's called dolomite. It's similar to limestone. Limestone is a carbonate rock, and caves tend to form in carbonate rocks. And so all of these are ancient caves that have been filled in with sediments preserving fossils. This is a picture of the site as it stands today. Now, I will suggest to you that this doesn't really look like a cave, right? It just looks like a hole in the ground. And in fact, what it is, a hole in the ground. It's a circular or cylindrical hole. Here we're looking at the western side of that cylinder. There is the north side where the people are standing, is over to the east. And then the south side is going to is sort of behind the field of view of this picture. And this sort of gray blocky walls here represent the dolomite, which is the bedrock in which the cave formed. And these sort of brownish sediments here with cobbles in them or rocks, these represent sediments that have fallen into the cave and then also contain the fossils that we're interested in. Now, the cave didn't look like this 2 million years ago. This is what it looks like today after a lot of erosion. I'm going to show you a schematic cross-section of the cave. It'll be a cross-section that'll go from east on the right side to west on the left side. And so here is the east side of the cave where those people were standing. Here's the west side. And you can see that about 2 million years ago, there was a large domed roof over the cave. And in fact, it's shown here with an opening, a chimney up to the surface. But when the cave originally formed, actually, that chimney wasn't there, and it was just a cavity underground. And the way the cavity forms is that water dissolves the rock. Carbonate rocks are water soluble. Water dissolves a cavity inside the rock. Water levels recede, leaving an empty cavity with nothing in it. Water then filters through the rock. Rainwater, for instance, filters through the rock. Um, as it goes, it's dissolving a little bit of the dolomite, picks up calcium carbonate, which then drips into the cave, forming what are broadly known as dripstones, or you may be familiar with stalactites or stalagmites. Water drips down here, leaves behind a precipitate of calcium carbonate, falls down onto the floor. And actually, the first thing that forms inside the cave is this broad, flat sheet of calcium carbonate. This is known as a speleothem or a flowstone. It's going to be important to the story in a moment. But then eventually erosion opens up a vertical chimney and stuff starts falling in. What falls in? Rocks, dirt. If there are bones from animals that have died on the surface, they can fall in. Sometimes animals themselves can fall in, die in the cave, and their skeletal remains will be left behind. So what you find at a certain point inside this cave, as it fills up over, tens of thousands or hundreds of thousands of years, is you get dirt and you get rock. There's water because it's still raining and dripping through the roof and coming in this hole. And then that water dissolves the dolomite, producing this calcium carbonate precipitate. And when you combine dirt, rock, water, and calcium carbonate, you get cement. You get a natural cement. That cement is called a breccia. It forms around the fossils and then preserves them very well, meaning that you have a chance to find those bones millions of years later. How do we find the site? That finding the site relates to this deposit of flowstone at the bottom of the cave. So over 2 million years, the roof of the cave would have eroded off, and it would have been mostly filled with sediment here. But this flowstone here becomes very important to miners. So the site was discovered because of mining activity. They weren't mining for limestone, or they weren't mining for gold or anything like that. They were mining for this stuff, this calcium carbonate. The reason why is if you superheat calcium carbonate, it turns into something called lime. And that becomes an important product that is used in gold mining. It helps separate gold from this sort of flakes of gold from this sort of slurry of mud that comes out of gold mines. And so it was a very important product used in the gold mining industry in South Africa. And so miners came here, they saw the calcium carbonate, just blasted it out. We're trying to extract it out, and they left behind this column, this mound of breccia here because they weren't interested in it, and just took out the calcium carbonate along the bottom. We know the miners were there. Before I get to that, actually, how do we know that the way I've described the formation of the cave is accurate? How do we know that stuff fell down a chimney and formed a mound? We know that by looking at the sediments. So I want to draw your attention to this sort of pinnacle or column of breccia right over here, and we're going to pay close attention to those rocks. And you can see if you look at those big clasts of rocks, they are actually organized in layers. And this was the original slope of that mound of dirt that fell in through the chimney. And so this is telling us that the opening to the cave was off here, up to the right, and then off to the screen above, somewhere in the center. But it was closer to the western side of the cave than it was to the eastern side. So when miners came here, like I said, they were after this calcium carbonate deposit. We know they were there because they dug this enormous pit and lined it with brick. This was the kiln that they used to generate, they built an enormous bonfire here that they would then use to superheat the calcium carbonate and transform it into this material called lime, which was then sold to the gold mining companies. Now, a geologist, because miners often found many of the cave sites in this area that led to human evolution, led to paleoanthropologists coming to the region, a geologist began investigating the site in the early 90s. His name was Andre Kaiser. He came in 1992. They knew the kiln was here, so they knew there was mining activity. That suggested that there was a cave. They started he started scrapping around on exposed faces of breccia near the walls of the cave, and he found this fossil. This is actually part of the brain case of an ancient baboon. And so he knew, okay, there are fossils at the site. He's going to start working here. And he began excavating, and this is a 3D rendering of the site as it is today. There's a lot of topography because it's been a lot of irregular topography because it's been excavated. But at the time, it was just a gentle slope from these cliffs down here to the floor. And he began excavating in the center, and he got super lucky because very, very soon after he started excavating, these sediments had sort of decalcified. They were no longer cemented together. Rainwater from being exposed to the surface led them to sort of split apart. So they were just simple sediments he could excavate away. He came down on this large mound of still calcified breccia, and stuck to the top of it was this specimen here. This is specimen DNH7, which is Drimolen hominin 7. Boy, we have so much great stuff here. We've got a relatively complete neurocranium. We've got a face. We've got a mandible. You can see most of the teeth here. And we know from details of principally facial anatomy and also the really large molar teeth, large premolar teeth, but very small incisors and canines, and the really, really large mandible, that this represents an individual of an extinct hominin in the species Paranthropus robustus. And in fact, this is the most complete skull of Paranthropus robustus known. And we also know from its very small size and also some details of the facial and skull anatomy that it very likely represents a female, which was great because at the time it was discovered, there was some ambiguity about which specimens in Paranthropus robustus were in fact females. Here is sort of a picture of the specimen more or less held in place, and you can see it's an absolutely lovely individual, and there's lots of information you can get from this. So this was amazing. It put the site on the map. There was then excavations that took place over, a couple of decades. And they found stuff. They found like teeth, but nothing else really quite as spectacular as this specimen. Now, they were excavating mostly in this area here, which is called the central excavation area, where there were decalcified sediments that were easy to excavate. But it's possible that that any spectacular fossils there were essentially dissolved and were lost. And so you mostly find isolated teeth. And then when our team took over the site about 7 or 8 years ago, we began excavating farther back in areas that were only partially decalcified. So there hadn't been a long time since the fossils were freed from the surrounding breccia. And in this area right over here, just to the south of this pinnacle where we found DNH7, in this spot, sort of coming out of a wall, we found a really amazing specimen. It's also probably just about 2 million years old. There's a lot of the dating of the site is really well done, and we think we have good control and a good understanding of how old the site is. So at about 2 million years old, we found another specimen, DNH 134. It originally came out as about 50 pieces, but when put back together, it is this absolutely beautiful neurocranium, or part of the skull enclosing the brain of a child. This was a young individual, probably between about 2 and 4 years old when it died. And there are just a few subtle characters here, like this flattened area here on the back of the skull, and the very sort of pronounced curvature or angulation in the back, and details about some bony sutures. They're quite pedantic, if I can just say. But because of some of these features, we infer that this thing is probably Homo erectus. And in fact, it represents the oldest Homo erectus skull, chronologically oldest Homo erectus specimen in the entire world, at just older than about 2 million years ago. Homo erectus, it turns out, is a super important species for understanding human evolution. This is a very simplified family tree of hominins, like I said, of human ancestors. And Homo erectus occupies this central position here. It's important because it lived for a very long time. It's arguably the first hominin species known to have dispersed out of Africa. It is very likely broadly speaking, an ancestor to almost all of the other members of the genus Homo that follow it, including us. So it's probably one of our direct ancestors. And something that's really neat about it is it lived for such a long period of time. There's actually a period of time for about a few 100,000 years when Homo erectus is, at least as far as we know, the only hominin alive on the entire planet. So right now we are the only human species alive, but Homo erectus was like us before we even existed. It was really quite a remarkable species. There's also some really amazing stuff about Homo erectus that speak to its paleobiology that relates to our own evolution, and that has to do with brain size. So what we're looking at here is a plot of fossil hominin specimens. This is on the bottom a time scale from the present day on the right to about 3.5 million years ago. And all these symbols represent individuals of different types of hominins and different species. And it looks like, oh, well, brain size increases over time. ECV stands for endocranial volume. So this is cranial capacity measured as either milliliters or cubic centimeters. And it looks like, okay, human evolution is characterized by an increase of brain size over time. But if you look more closely at the data, there are interesting patterns that emerge. So this is the brain size that you might expect to find in a chimpanzee and also in the pre-australopiths. So this is sort of a baseline of where brain size was when human evolution starts. By the time you get australopiths, you get a step up in brain size. It's about 20% larger in the various australopith species. But you can see that pattern is that there's a flat, there is no trend. The relationship between brain size and time is relatively flat. Then we see this large increase. This corresponds to evolution within the genus Homo. Okay, that's great. But if you look more closely at it, up at the top here, you find more recent hominins like Neanderthals and modern humans. At the bottom here, you have things like some of the very earliest members of Homo, like Homo habilis. But if you look at Homo erectus, you can see that Homo erectus is actually the species, the lineage of Homo erectus over time is actually responsible for turning members of the genus Homo from, relatively small brain creatures to actually quite large brain creatures. And so understanding the evolution of brain size in Homo erectus is really important for understanding why our own brains are so large. And now, where does our specimen DNH134 fall? Well, it falls down here towards the bottom. It's very, very chronologically old, but it also has a relatively small brain size. And so probably in this period right here is we're going to pick up the earliest evidence of brain expansion in this very important Homo erectus lineage. Now, that's not the only thing we've discovered at the site. We talked about discoveries down here, but back over here in this sort of like another little sinkhole, a little dissolution cavity up against the wall of the cave, we have found another specimen, DNH155, and I'm going to show you the site. Imagine you're standing on this platform over here looking towards that area. This is what you'd see. There's a little depression down here behind the sort of wall of breccia. You can see the people working back there. And there was a field school student working back there, Samantha Good, who was been back there for a week or two. And then one day she sort of poked her head over this wall and said, Angie, one of our project co-directors is Dr. Angeline Leese. Angie, could you come over here and take a look? I think I found something. And so Angie climbed over this wall of breccia, went down below it, and then popped up like a jack in the box and told everybody to come over. And this is the moment of discovery here. There's Samantha Good. There's my colleague, Angeline Leese. These are my colleagues, Jesse Martin and Stephanie Baker. Oh my God. Sorry, Steph. I just had a momentary lapse. And we're all looking down here. And what do we see? Well, over on the left side, up against the wall of the breccia, where it's just starting to decalcify, we find this row of teeth, and this is the roof of the mouth. This is the palate. So imagine it's the specimen is lying sort of upside down here, and we were super excited, finding this is a relatively complete fossil. This is great. We were very excited. Keep it covered. So when we find cool things at Drimolen, we dance. I know you're all very impressed by my sick moves. But anyway, we continued to excavate, and we dug down a few more centimeters, and then it became clear that, oh, we are picking up more of that specimen here. This is more of the cranium. This right here is the foramen magnum, which is the hole on the bottom of the skull through which the spinal cord leaves the brain, and goes down into the vertebral column. We took this thing out as a as a block, sort of consolidated with some glue that can be dissolved in acetone. So we held it all together. The fossils that come out of Drimolen can be quite fragile. We then painstakingly cleaned it off, and when I say I mostly mean my colleague Jesse Martin. Here you can see that that was the original set of teeth that we discovered in the original palate. As we exposed, we saw that there was basically the face had sheared in half and slipped down. Here's the other half of the face emerging with its teeth. A nice sort of cleaner shot of the specimen after it's been cleaned off. Here are the two halves of the palate put together. You can see we have all the teeth. It's an old individual. The teeth are highly worn. You can see the very large size of the molars and premolars, the comparatively smaller canines and incisors. That's a hint as to what it might be. And we kept cleaning off and putting things back together, and we emerged with this thing, which is not the most complete, but the best preserved cranium of Paranthropus robustus ever discovered. It's really amazing. It and that specimen DNH7 have very, very subtle anatomical features that differ from Paranthropus robustus specimens that are chronologically about 100,000 or 200,000 years later. And so what we think we're picking up is of evidence of transformation within the Paranthropus robustus lineage over time. That's a really neat evolutionary phenomenon that's terrific to document. Also, DNH 155, this new specimen, is really very broadly the same age as specimen DNH7. They're also from geographically exactly the same place. And so it's likely that they represent a male and a female. There are some other characteristics of this specimen that indicate that it's male. It's slightly larger size. There's some cresting on its cranium that suggests it's a male. We're probably picking up males and females from really, broadly speaking, the same population. And so we're going to be able to really drill down on that and eventually reveal really interesting details of the paleobiology of this population of this species existing at this particular time. And so to summarize, Drimolen preserves the best evidence of Homo erectus in South Africa and the earliest evidence of Homo erectus in the world. Drimolen has produced the most complete skull, DNH7, and best preserved cranium, DNH 155, of Paranthropus robustus. And fossils from Drimolen are giving us insights into the paleobiology of our ancestors at a time when members of our own genus Homo begin to emerge and acquire increasingly human-like characteristics, including large brains. So I'd like to thank my colleagues, the field school participants who every year, they change every year, but they're the ones who do a lot of the work in recovering these fossils, our institutions, the University of Johannesburg, Washington University in St. Louis, as well as La Trobe University in Australia. And I'd like to thank the Australian Research Council, who has funded our project very generously. And thanks to all of you for coming.
That was really fun. I particularly enjoyed seeing those like moments of discovery photos. So thank you. That was fantastic. We're going to launch right into the Q&A. So we have some questions already. And the first question from Steve is, how was the landscape and environment changing around Drimolen Cave when Paranthropus robustus and Homo erectus lived?
Oh, that's such an excellent question. Drimolen Cave is really, really interesting in terms of its paleoecology. So we know from other sites in that same region of South Africa, those other little dots on that map, that sites that are a little bit older have sort of what's called a mixed environment or mesic environment. It's a little bit wetter than what you might see today, for instance. There's more bushy cover, more trees and stuff like that. And then we know from sites a little bit younger than Drimolen that the climate and the environment is much drier, less tree cover, more open savanna. And so there is this transition that takes place in southern Africa from a slightly wetter to a notably drier
Mhm.
climate. And Drimolen Cave is found in terms of its chronology right in the middle of this transition. And so we get cool things that happen at Drimolen Cave. So for instance, one way that you can tell the climates are and the environments are different at older or younger sites is that the fauna that's preserved are different. And so there's a turnover in the types of antelope or the types of predators that are present, 2.5 or 2.2 million years ago, versus the same types of animals that are present 1.8, 1.7 and younger. And Drimolen's about 2 million years old. And what we find is we get some of those early fauna persisting up until Drimolen, and then some of those new fauna appearing at Drimolen. And so Drimolen is really picking up a moment while that faunal turnover is taking place. So
Nice. So
all
going from
that
sort of more,
environmental change is really important for understanding the adaptations of the hominins that
and it
live
sounds like in general is going from sort of wetter, more mixed to drier in general. So, okay,
exactly.
cool. I'm going to go to one of our behind the scenes questions. And so, if DNH7 and DNH155 had been found at different sites, what do you think the chances are they would have been described as different species?
So that's a really interesting question. Um, there might be reasons to suspect that this happens in paleoanthropology from time to time. In this case, I don't think that would be the case because
Mm-hmm.
um, DNH7 and DNH155 share a ton of features. They're
Mm-hmm.
different in size, and there are differences that are characteristic of males and females in the same species when there is this phenomenon called sexual dimorphism where males and
Mm-hmm.
females differ in size and shape. But there are a lot of very, very small scale features that are similar in DNH7 and DNH5 that differ from hominins discovered at other sites. So I'd like to think
Mm,
that even
yeah.
if we found those specimens at different sites, we would associate them together.
Okay. This is another question from Steve in the audience, which follows on that very well. So he asks, were all robust forms of Paranthropus robustus and early Homo only males, or do you see robust forms in males and females? Do the robust forms reflect environmental factors, not just sex?
So when we refer to robust species of australopith, we're talking about species that in general show amazing adaptations in their faces, in their chewing muscles, in their teeth that suggest that they subsisted on a diet of foods that were hard to process. There is actually an ongoing debate of which I'm part of, about whether or not these things ate very hard foods or very tough
Hmm.
foods or something like that. But there is a sense that they have these amazing adaptations for feeding on foods that are difficult to consume.
Hmm.
Um, and so those robust features are present in both males and females. So the
Hmm. Okay.
robusticity of the specimens is not
Hmm.
simply that the males are bigger; it's that they
Hmm.
have shapes to their skeletal features that really set them apart from other types of hominins or other apes. And I'm not sure if there was a second part of the question.
No, I think that's good. Yeah. Robert asks, do you think the early evidence of Homo erectus in South Africa indicates that the species evolved there rather than in East Africa?
Um, so that is really an interesting question. Um, so if you're thinking about robust australopiths in general, uh, the chronologically oldest evidence of robust australopiths is in East Africa. And there is evolution that takes place in East Africa, and you get this transition in terms of a sort of a slightly less derived to slightly more derived type of robust australopith in East Africa. Now, the question, does that chronology mean that robust australopiths evolved first in East Africa?
Actually, the question was about Homo erectus, not.
Oh, I'm so sorry.
That's okay. Yeah. But same
Um,
question, basically.
Same question though.
Yeah.
So, no, I wouldn't put too much emphasis on the fact that we have an early date in one particular region.
Mm-hmm.
That could just be a sampling phenomenon. We actually
Mm-hmm.
have a fragment of a Homo erectus cranium from Eastern Africa that's just a little bit younger. It's a little bit young. It's like 1.95 or 1.9
Mm-hmm.
years old. So that's about the same time. So when you find early evidence of a species in one region of a continent, that doesn't necessarily mean that that region of the continent is the site of origin of where the species first evolved.
Mm-hmm. Yeah, thank you. Agreed. So Urate asks, did I miss it or was there any postcranial material discovered at Drimolen?
There is some postcranial material from Drimolen. Um, it's scrappy.
Mm-hmm.
There's a pelvis. There are quite a few finger and toe bones, stuff like that. There's not really great postcranial evidence, at least that has been described yet. Um,
Mm-hmm.
and then, of course, there is the challenge of when you find such material, being able to figure out what species they belong to, because we've got multiple hominin species
Mm-hmm.
at the site, and most of the taxonomy is based on skull or dental or cranial or mandibular traits.
Mm-hmm.
Um, and so if you find an isolated femur, with a thigh bone or something like that, it can be a little difficult to attribute those things to different species. Some of the really exciting things that are happening in paleoanthropology now, in general, is that we're beginning to find an increasing number of specimens where postcranial material is associated with cranial and dental material, and so then you can start to match up, the body below the neck with the body above the
Mm-hmm.
neck, and begin to make firmer taxonomic conclusions. But right now, we don't have a ton of postcranial material.
Mm-hmm.
We're hoping to find some more, and we're hoping it's going to be associated with like jaws and teeth.
That'd be great. Is there for the postcranial, because that was going to be my next question, has any of the postcranial material been given any taxonomic IDs or it's still just hominin really at this point?
Um, Even at other sites, you could at Swartkrans, where you've got all this, you have a
Mm-hmm.
more extensive record of postcranial stuff. It can still be sort of hard to find
Mm-hmm.
identify differences between, say, Homo or Australopithecus and
Mm-hmm.
Parapatus. It's usually at the level of genus.
Yeah.
Um, there are some parts of the body where you might expect to find um, bigger differences in form,
Mm-hmm.
but uh, I'll say that it's mostly hominins, and
Yeah.
we might think a hint of this might be Homo, this might be Paranthropus. Nothing we're super confident in.
Okay, that's fair. So Lynn asks, is there any DNA or even protein evidence from the site?
Well, this is super fun. So, no direct DNA evidence. I gather, Lynn, that it has been possible to extract proteins from fossils that might be 2 million years old. And in fact, there are some reports of proteins being extracted from teeth at other sites in South Africa. And we are working with the team who is thinking about this issue. And so we may be able to extract proteins in the future, but we haven't done so yet.
Cool. Fingers crossed. You can come back and tell us about that.
Yeah.
I'll go to one of our behind the scenes questions. So since early Homo, like the oldest Homo erectus, are not as numerous as Paranthropus at these sites,
Mhm.
do you think that reflects any kind of real life difference in population numbers or abundance, or is this more likely due to a difference in ecology or behavior, or just some sort of sampling bias or taphonomy, I would add at the end?
I think it's hard to say for certain.
Mm-hmm.
But what's interesting is when you look across East African and South African sites,
Mm-hmm.
at most sites where you find Homo and Paranthropus, you find that Paranthropus overwhelmingly outnumbers Homo. in terms of the representation of the fossil record. And those represent very different um, depositional environments. I mean, the one
Mm-hmm.
site where I guess there's an exception to that is Olduvai Gorge, where it's closer to a 50-50 split. But most of the time, it's like most of the fossils are like 9-1 in favor of
Hmm.
Paranthropus. So, you know, I wouldn't take a firm position, but I think it's a reasonable hypothesis to think that maybe Paranthropus was actually more numerous on the landscape. It's possible.
Interesting. I never thought about that. So I'm going to do another behind the scenes question. So Drimolen allows you to glimpse into the microevolution of a single lineage, which is very cool. Do you think this potential currently exists at other sites with other hominin taxa? Or does it seem like other species are more sort of monolithic in their morphology through time? They just, that morphology stays the same. Yeah, are there
Well,
other places where you might be able to look for this?
well, so I don't think it's within a site is a little tricky, but if
Mm,
you look across stretches of time in the same geographic area, I mean, I think you've got really compelling evidence that in Europe during the Middle Pleistocene, you have local evolution from some type of sort of post-Homo erectus, archaic Homo thing. Sometimes that
mm-hmm,
stuff is called Homo heidelbergensis, and
mm-hmm,
that population evolves sort of in situ in Europe and turns into Neanderthals, and that
mm-hmm,
is microevolution. It's anagenesis. It's change within a lineage.
mm-hmm.
Um, that's a really good example.
Mm,
In East Africa, there has been talk about transformation within a lineage from material that traditionally is called Australopithecus anamensis up to Australopithecus afarensis,
mm-hmm,
and this represents a change within the lineage. There have been some discoveries that have now challenged that idea, but I think it's still
mm-hmm.
an idea worth thinking about. Um, uh, there if you look at there are other sites in East Africa with a deep stratigraphic sequence where you maybe find some evidence of some features changing, but then other features not really changing. So I think
Mm-hmm.
you're not going to expect to see it every time. And so I'm not
Mm-hmm.
sure if I can generalize about
Mm-hmm.
how frequent stasis versus gradual
Mm,
evolution is in the hominin fossil record, but it is certainly a phenomenon that I think can be observed.
cool. Since you mentioned Homo heidelbergensis, we have a question from Dudley who asks, you show heidelbergensis as an ancestor of modern humans. Chris Stringer of the UK Natural History Museum rejects this. Do you disagree with him? And if so, can you explain the controversy, as he said?
Sure. Chris Stringer will be very amused to hear me pontificate about the evolution of later Homo, but I'm going to do my best here. Let's put aside the issue about taxonomic names or labels and stuff
Mm-hmm.
like that. What you find, starting about 600,000 years ago and a little bit younger, is you find the appearance of these hominins that differ in subtle ways from Homo erectus. The brains are a little bigger, the cranium's a little bit rounder. There are some distinctive features in the face. The brow ridges are very distinctive in these things. And these hominins show up all around the world. They show up in Africa, they show up in Europe, and they show up in Asia. And one school of thought is that, wow, these things all mostly look the same, um, even though they're in different parts of the world. Um, Maybe it makes sense to treat all of these things as if they were a single species. Where we're a single species, we live all around the world. Um,
Mm-hmm.
so that's not impossible. And if that's the case, then you would call those things Homo heidelbergensis just because of an accident of history of when the
Right.
first fossil was discovered and named. There is another school of thought that what we're picking up here is we're getting divergence of um, different sort of regional populations, and they're splitting away from each other. And
Mm-hmm.
so in Europe, that population of these archaic humans is already diverging towards Neanderthals. The population in Africa is diverging from the Asian and European populations, is eventually going to give rise to modern humans. And then there's a population um, in Asia. Um, and if that's true, then the African population gets given its own species name. The Asian population gets given its own species name. I'm inclined not to go down that road, and I prefer to think
Mm-hmm.
of them um, as um, as sort of one likely
Mm.
coherent population. You,
Okay.
you can perhaps identify little differences between them, but I don't think they're super profound. So
Okay.
that's my personal view. I could go on and on about this, but then I would be going well outside of my comfort zone. So less Chris Stringer see this and then send me an email telling me.
Maybe we'll have a conversation at a conference
Oh
at
yes,
some
there we
point.
go.
That'd be
Chris and I
fun.
are friends and stuff, so it's
Excellent. So since you also mentioned before in the answer to the question before this, you mentioned Australopithecus afarensis. And so one of our behind the scenes questions is that the timeline you showed has Australopithecus afarensis going back to 5 million years.
oh.
Do you think that that's a long-lived lineage? Where do you get those older dates from?
So that phylogeny that I showed you, like I mentioned that it was simplified,
Yeah.
and actually it's not just simplified. What it is that's a phylogeny that's in a recent paper of ours that was published in 2024,
Hmm.
talking about um, species concepts
Hmm.
and how we identify species in the human fossil record.
Hmm.
And the conventional way that paleoanthropologists identify species is by considering basically morphology and variation. So if you measure variation and you find a bunch of things that look the same, sort of the same, and they
Hmm.
vary from each other, but within an amount of variation that we think is appropriate for a single species, then we call it
Hmm.
its own thing. And so basically, if it if the specimens look like each other and they look different from
Yeah.
other things, then we give them different species names. And that's, to be technical, that's an idea called the phylogenetic species concept.
Hmm.
And it has been ascendant in paleoanthropology for the last 30 years at least. And my view about that is that that's probably not the best way to think about species, and that species are, if you think about not how you identify species, but what a species really is, a species represents a lineage of ancestors and descendants that have diverged from other groups. And if you
Hmm.
think it's a real phenomenon that within a lineage you can observe evolution, that means that early members of the population, early members of the lineage may differ morphologically from late members of the lineage,
Hmm.
which means that under the phylogenetic species concept, those things would all of a sudden become separate species. But you haven't
Hmm.
actually falsified the possibility that one is just a linear descendant from the other. And so that phylogeny that I showed you and the species there is highly conservative. And so the early
Hmm.
dates there would include things like Anamensis and even some of
Ah,
the earlier fossils, because we feel like we can't falsify the idea that those things that we can't demonstrate that lineage divergence has taken place in those things. And so, for instance,
got it.
going back to sort of the Anamensis afarensis example that I use, most people consider those two things different species, but it has been argued, even by people who call them different species, that they're just a lineage. And if they're
Hmm.
just a lineage, there's actually no reason to call the early part of the lineage a different species. And so I should say to the audience that my views on this are probably it's a minority view. Many of my colleagues disagree with me, and that's fine. That's how science works. But I think I'll just go and say that I think paleoanthropologists need to be a bit more introspective about how we go about defining species, and I think we need to think a little bit more critically about it.
No, that's great. That's really helpful. Um, Another audience question from Urate. Were Paranthropus and Homo erectus suspected to have totally different diets at this time, sort of going back to Drimolen?
Um, so that's a complicated question to answer.
Hmm.
Uh, it wouldn't be surprising if aspects of their diets overlapped. You
Hmm.
can find today there are sympatric populations. Sympatry means living in the same place in the same area at the same
Hmm.
time. And you can find populations of chimpanzees and gorillas occupying the same forest, having overlapping ranges,
Hmm.
and they will feed on some of the same foods. They then also diverge in other aspects of their diet, and it wouldn't shock me if Homo and paranthropus are sort of doing the same thing, overlapping in certain aspects of their diet, but also diverging in other ways. We know that in the genus Homo, well, we know in australopiths over time, there is a trend towards increasing robusticity in terms of the molar teeth, the thickness of the enamel, so how strong the teeth are, the size of
Hmm.
the chewing muscles, the size of the jaws. Those things are all getting larger and larger and larger over time. And then at a
Hmm.
certain point, Homo departs from that trend, and then all those things become smaller and smaller and smaller. And so it does suggest that there is a dietary difference.
Hmm.
I wouldn't say that it's necessarily an exclusively non-overlapping differences in diet.
Okay. Yeah. And she just asked in the follow-up, she said maybe teeth isotopes would be informative of diet differences.
Yeah, but what you find is that uh, there's a cool pattern also that over time in australopiths you get an increasing amount of um, evidence of C4 plants. C4 plants are
Hmm.
plants like most grasses have a particular type of photosynthesis that winds up leaving an isotopic marker in the teeth. Um, and then the animals that eat grasses also pick up that signal, and so animals that eat the animals pick up the same signal. And
Hmm.
what you find in Paranthropus robustus is that there's a lot of variation.
Hmm.
That some individuals would have had a very high proportion of their diet derived either from those C4 plants or from the animals that ate those plants.
Hmm.
But other individuals don't, and other individuals in the same population would have had would have relied on what are called C3 plants, which are plants with a different type of photosynthesis that include trees and bushes and the products they
Hmm.
produce, like leaves and berries and fruits and stuff like that. And then the
Hmm.
animals that eat those plants. And so some individuals in Paranthropus robustus would have eaten a lot of browse, fruits,
Hmm.
weeds, or the animals that ate those things. And so you can find even within this one species, there's enormous variation.
Hmm.
And I think that's people tend to think about the isotopes as like, oh, well, these things ate grasses or these things ate
Hmm.
browse. But I think actually the really cool thing in those data is the variation within even a single species.
Nice. The in the answer to the question before this, you talked about, Homo diverging from a trend, and so this question follows on nicely. Another question from Irate: where does the size of the cranium of Homo naledi fit on your time chart? I think of brain size is what the yeah.
So Homo naledi is actually just a little star way off on the side here, drops down
Mm-hmm.
low. Homo naledi is a relatively young um,
Mm-hmm.
species, and it has a reasonably small brain.
Mm-hmm.
Not like super duper small, like Homo floresiensis, which is another
Mm-hmm.
one of these small-brained individuals, but it's going to be on the smaller side. So Homo naledi and also Homo floresiensis are sort of outliers to that
Mm-hmm.
overall trend.
Okay. I've
These are
two.
amazing questions. Can I just say, wow,
And we have two more. I think we have time for two more questions that we have
this is
from our
like I feel
behind the
like I'm being
scenes team.
quizzed. Okay, yes.
And so one is uh, the term Australopith, which you've been using a lot, is sometimes criticized for suggesting that there is a tribe, Australopithecini, and that might be a real, okay, hold on, a real and valid taxonomic group. And so the phylogenetic data don't necessarily suggest this. Do you think this term Australopith is useful despite this technicality? Is it more of a sort of not a taxonomic term, but a way that anyway, go ahead.
Okay. So, um, so there's a difference between the term Australopith and Australopithecine.
Yes.
So when
Okay.
I was a graduate student like 30 years ago, we still used the term Australopithecine. And
Mm-hmm.
Australopithecine is an informal expression of, um, of a subfamily name, the Australopithecinae.
Mm-hmm.
Um, and, uh, and that implies the existence of a taxonomic group and under a certain way of thinking, a way of thinking to
Mm-hmm.
which I also agree with. That suggests that that should be a monophyletic group, a group of animals
Right.
all descended from a single less common ancestor. And we know that that's not true. We know that Australopiths do not form a clade. They branch off
Mm,
separately off of the base of the hominin tree. And so Australopithecine sign is not a proper term to use because it implies the existence of a formal
yep.
subfamily. But
Mm-hmm.
Australopith
Mm-hmm.
does not because it doesn't have
The
that suffix
taxonomic
at the
part.
end.
Yeah.
And so
Mm-hmm.
it's a general term, it's a
Yeah.
gradistic term,
Mm-hmm.
and it's a useful shorthand, but that does not have the disadvantage of implying the existence of a formal taxonomic group that doesn't exist. And so folks who work on early hominin evolution have over the last, I don't know, 10 or so years, maybe 15 years, started using the term australopith instead of australopithecine to be able to still
Mm.
refer to the same group of animals, but not have that taxonomically incorrect implication. So an excellent question.
Okay, great. Thank you. And okay, our last question before we wrap up, I think, is what brought you to Drimolen in the first place, and where did your passion for interest in South African paleoanthropology come from?
How much time do we have? We have a few minutes?
We have a few minutes, yes.
Okay, my involvement in the Drimolen research team is just one of these amazing um, contingencies of life. So I they were finding amazing stuff at Drimolen, and the team was looking for folks with expertise in sort of early hominin cranial dental taxonomy and systematics and stuff like that. And there were some graduate students interested in this, and they were wanted to bring on a senior member of the team to help advise those graduate students.
Hmm.
And one of those sort of very early career, then younger graduate students was that person I mentioned, Angeline Lees, who had gone to graduate school in Australia. But before she went to graduate school in Australia, Angie was my undergraduate student. And and I had also known some other people on the team a little bit. And so in those discussions, the team settled on asking me. And I was delighted to join the team. But so there, you know, it's there are all these little human elements to and social relationships that help to determine the course of people's careers. And so this is a wonderful opportunity to me, but it also arose because of circumstances involving the development of people you know. So that was a lovely story. My interest in human evolution in general, not just in South Africa,
Hmm.
was that I'd always been interested in evolution as a kid. I grew up in New York City, used to go to the Museum of Natural History, but I didn't really think about paleoanthropology as something I could do until I went to college and I took a class in it. And I thought that was amazing. And I decided, oh, I want to major in this. Maybe I want to go on and be a professor or something like that. And I went to see a graduate student who was living in my dormitory, who was an anthropologist. This is Dan Lieberman, Briana, if you
Hmm.
know. He's a professor at
Yeah.
Harvard now. And he said, oh, well, you should. And I went to him and I said, hey, I'd like to do this, but I'd like to know if I can go into the field maybe and get some experience. And he said, oh, well, you should talk to my advisor. And his advisor was Ofer Bar Yosef. And are we going to get cut off in one
Nope,
minute?
no,
Can I
we're
go a
not.
couple more minutes?
Yes.
Um, so Dan set up an appointment with me and Ofer. And ordinarily, when an undergraduate student goes to see a professor, they go to their office hours or go to their go to their office and set up a meeting and stuff like that. But Ofer came down to my dormitory and had lunch with me in my dorm. And then I'd never met him before, but he said, "Okay, you want to be an anthropologist? You want to be an archaeologist? I'll send you to France." And and he arranged, like within the first half hour of meeting me, he arranged. I'd never taken a class with him. He arranged for me to spend 6 weeks in France digging at a Paleolithic site with one of his colleagues, and I went and I loved it. And then on the weekends, we'd get a little break. I would go into Paris, and then um, I met my wife at a bar in Paris.
Oh,
So life
lovely.
life is what happens to you when you're like doing other things.
And I think both of those stories really just speak to the importance of mentoring and teaching. So
Yes, exactly.
that's a great way to wind up. So thank you. So we will conclude today's virtual program. So please join me in thanking David for sharing his work with us. I'd also like to give special thanks to those who made this program possible, to our behind the scenes team who helped sort through your questions, including Dr. Ryan McRae, who works with us in the Human Origins program, to our donors, volunteers, and viewers like you, and finally to all our partners who help us reach, educate, and empower millions of people around the world today and every day. We thank you. This is our last webinar for this year, but stay tuned and check your email for information about the next season of Hot Topics. We will resume in February 2026, and we will send information about that webinar soon. We'll put a link in the Q&A where you can find information about our upcoming programs. We just did that, and how to sign up for the museum's weekly e-newsletter. That is the best way to stay informed about upcoming programs and learn more about the museum's research and exhibitions. So right after this webinar ends, you'll see a survey pop up asking for some feedback about the program. Please take a moment to respond. We're very curious to know what topics you might be interested in seeing for future programs, and we appreciate your input. So thank you, Dr. Strait. Thank you to our participants. Thank you to the audience, and we will see you next year.